Good morning, good afternoon, depending on where you are. Welcome to this webinar. My name is Heike Schmidt. You see me today in my role as Senior Advisor to the Wetsus Water Technology Institute. I also work at the Dutch National Institute for Public Health and the Environment and at Utrecht University. It's my pleasure to host you through this afternoon, which will consist of a couple of presentations on AMR, antibiotic resistance in wastewater, and in wastewater treatment plants. From these plants, resistance gene and resistant bacteria can reach the environment on a daily basis. And plants have been discussed to be a hotspot of antibiotic resistance, probably for the spread of resistance between bacteria and for the spread into the environment. But actually, they could also be points where we can intervene and that help us in reducing emissions to the environment. Therefore, in this afternoon, we will be having presentations about the presence of antibiotic resistance in wastewater treatment plants, what determines their presence and their reduction in these plants. We'll also look at how genes move between bacteria and finish off and the presentations with a presentation on a applied view from a water board. We'll start with the UK and then move to the Netherlands, where one of our PhD students within Wetsus has conducted her experiments. That also brings me to the organizations that supported this webinar to come true. That is, first of all, the EU-funded Repares project, Horizon 2020 project, and also the Dutch Foundation for Applied Water Research, STOA, has contributed to the PhD thesis of Rebecca Palaris. Our invited speakers consist of Professor David Graham from Newcastle University, and then Rebecca Palaris is announced a PhD student at Wetsus, and we finish off with Maarten Nederlof. A few housekeeping rules. You can post questions at all times. And how can you do that? Just by typing them down and sending them to us throughout the presentations. We'll have room for questions after each presentation, and there's also quite some room for common discussion at the end of all presentations. So let's get going. It's my great pleasure to introduce our first speaker, who is Professor David Graham from Newcastle University. In his group, methods from engineering, theoretical ecology, as well as mathematics and biochemistry and molecular biology are all applied to answer pressing questions, one of them being the fate and transmission of antibiotic resistance through the environment. David, floor is yours on a talk about the spatial ecology of AMR in wastewater networks. Looking forward to your talk.
Great, thank you, Heike. We've been working in AMR, in wastewater and other environmental systems for about 20 years. And what I decided for this talk was to summarize results that we've observed over the last 2 years. And it's particularly results related to the ecology of drug resistance within treatment plants and wastewater networks. And as I hope I'll be able to convince you as I speak, you'll come to an understanding that ecological factors may be really important and may actually even drive what you find released from treatment plants and moving into the environment. So the talk is going to basically be a summary of just some general thoughts to do with antimicrobial resistance and the complexity of understanding it at the beginning. But then I'm going to go through 4 small subtopics that relate either directly or indirectly to wastewater, from wastewater networks from an ecological perspective. And I'll describe that first. And it's sort of the mentality that our group at Newcastle has developed over the last 3 or 4 years for explaining a lot of phenomena we couldn't explain in the past. Then I'm going to talk about the ecology of resistance genes and mobile elements within one particular wastewater network, and then we'll expand it out to other networks that we've studied. I'm then going to talk very briefly about mobile genetic elements, particularly class 1, clinical class 1 integrons. And recent data that we've got suggests that they may not be telling us what we think they are relative to markers of AMR, especially for modeling and monitoring. Finally, I'm just going to provide a little bit of laboratory-based work that we've done on gene transfer of biological processes. And at the end, through the summary, I'll talk about improving wastewater treatment plants to reduce AMR release. So I'm not going to cover a lot of background slides because I'm assuming a lot of people in this audience will know what AMR is and its context, but I'm going to provide a couple of things that are important for this presentation. So AMR is antimicrobial resistance, and resistance is basically the capacity of an organism to defend itself against an antibiotic or antimicrobial. There are two basic types of resistance: intrinsic resistance and acquired resistance. And intrinsic resistance, for our purposes, defined as resistance that we find in nature that is pre-industrial production and use of antibiotics, which is functionally about the 1940s. The issue of our problem of increasing antimicrobial resistance is really related to acquired resistance, and that's the gaining of resistance in bacteria after antibiotics have started to be used at an industrial and massive scale. And basically what we functionally done by increasing the use of antibiotics is increased the natural evolution of resistance. The problem is, by using them heavily in medicine and agriculture, we've forced bacteria to respond to the fact that we use them, and we now see bacteria, the wrong species, i.e. pathogens, collecting resistance in the wrong place. And one of the places where the consequence of that behavior is seen is in wastewater networks and treatment plants. So this acquisition process is primarily associated with the acquisition of resistance genes through either horizontal or vertical gene transfer. I'm primarily going to talk about horizontal gene transfer here. And so the question is, there's a number of different things we must understand, especially if we want to try and understand resistance from the context of waste treatment plants. So this is my general background slide, and basically it says AMR is complex, and it's driven by evolutionary, selection, ecological, and in fact, when it comes to waste treatment plants, engineering phenomena. It's rooted in the fact that bacteria want to defend themselves. So if we challenge them with a stressor like an antibiotic, they will rapidly respond through their SOS system and acquire, as they can, potential ways of defending themselves, which are genes. Once those genes have been acquired, they have an ecological advantage, and you have increased prevalence of the resistant strain. The problem is, from a management and decision processes, this is happening everywhere all the time. It happens in medicine, it happens in agriculture, and it happens in the environment. And as a consequence, it's very difficult to often track the cause and driver of resistance in any particular setting. And there is often an extended misperception that resistance was primarily transmitted and evolves at the point of use, because where we found resistance most significant was by MDs and vets in untreatable infections. So what we've got is a very complicated network of phenomena ranging from the original use to all the ecological environments in which the resistance genes and the bacteria must pass before they get to the environment and obviously to the next exposed person. Now, when we first were working on this, we had a whole suite of presumptions, and almost everything that I assumed, this was in 2005, I've changed my mind on. And what I want to emphasize in this talk is that some of the presumptions that we've come to believe may not necessarily be true. So what I'm going to do is I'm going to talk about wastewater treatment plants, which I think are the most valuable way outside of reducing the use of antibiotics, of reducing the spread of AMR. So I'm a great champion of wastewater treatment plants. But wastewater treatment plants are not perfect. And they were designed to remove carbon and more recently nitrogen, but they were never designed to reduce resistant genes or mobile genetic elements. Further, wastewater treatment plants are the endpoint of a wastewater network, which has got a diverse array of sources, and those networks are as diverse as the range of different ways you might pass waste from one place to another. So to understand resistance, and then develop ways of mitigating against resistance spread through wastewater systems, we've got to understand the ecology of networks, the genetics of resistance transfer, and also the engineering associated with different networks, which is really how we might make a network better for purpose. So to give credit where credit's due, most of the work I'm going to talk about over the next 15 minutes is by Marcos Quintela-Baluja and Florence Zhang. Marcos is a postdoc. He's a senior postdoc currently working for me. Actually, he's primarily working on COVID work right now, but he basically is one of our AMR genetics gurus within the group. Florence is now a postdoc at NUS Singapore, and the latter part of my talk is going to be about what work that Florence did when she was working as a PhD student for me. So this is my first and general overall slide. Is that when you think about this is how we started changing our thinking. We started thinking about wastewater networks as a series of ecosystems, and in reality, there's 4 characteristic ecosystems in a wastewater network. The first ecosystem is your gut. It's warm, it's organic. It could be the gut of a person who's in a hospital. It could be a gut in the person in the community. But the consequence of having taken the antibiotic, for example, and the resistance gained is the release of resistant genes in bacteria and genetic elements in fecal matter that ends up into the sewer line. And the sewer line is our second ecosystem. It's completely different than the gut. It's probably cold, it's dilute, and it's quite diverse in terms of the different types of environments you find there, because there's all different other sorts of wastes that are being put into the sewage as well. The third ecosystem is the actual wastewater treatment plant. But if you think about the wastewater treatment plant, it's actually not one ecosystem. Every single unit operation in a treatment plant is an ecosystem itself. The primary settler, the activated sludge unit, or the trickling filter, or the membrane reactor, whatever system you have for biological treatment, and the secondary clarifier. They're all different ecosystems that have got different chemistries and different drivers of survival of bacteria and bacteria associated with resistance. Finally, our final ecosystem is the environment, where the wastes end up going. It's also diverse. It's got smaller sub-ecosystems like the sediments, the water colloidal particles that are passing through the water. So in reality, what we're dealing with when we deal with a wastewater network and a treatment plant is a whole series of very, very diverse, chemically and temporally different places. And then you've got other ecosystem scale phenomena like the floc versus the biofilms versus the liquid phase that's not associated with floc and biofilm. Each of these places is going to have their own selective drivers for the organisms you're going to find there. And what I will show you is that in most of those locations, the presence or absence of resistance genes or mobile elements has nothing to do with the community that's actually there. It occasionally does, but it typically doesn't. And so what we've got to do is step back a bit and think about what's actually happening in a network and where the selection is occurring, and if there is any selection occurring. And we don't think there is much selection occurring in many places. So this is just to let you know, I'm only going to provide overview slides rather than going through lots and lots of details. But this is high-throughput qPCR data for a waste treatment network we studied in Spain. And there's a couple of things that I just want to flag. The first is wastewater looks like wastewater relative to the diversity of resistant genes. In this particular case, we had 2 hospitals, and we had 2 hospitals, HP1 and HP2, and we had community waste that didn't have hospital-associated waste. Well, the number of resistance genes and the abundance of resistance genes really weren't all that much different in the 3 sources. As I will show you, they actually are quite different. But the more important thing from a pragmatic perspective is what we see when we get into the treatment plant. And we compare the activated sludge itself, i.e. the floc, and the liquid phase effluent that comes out of the clarifier. The floc itself, and we've seen this now in multiple treatment plants, is actually the place where the lowest level of diversity exists relative to the variety of resistance genes you find. And the liquid phase and the floc phase in a clarifier are completely different in terms of their carriage of resistance genes and bacteria and also mobile elements. So you've got to first then think, well, okay, wait a second. This mixture we call activated sludge, which has got a liquid phase and it's got a floc phase. In fact, it's really two ecosystems that are operating in parallel. And what we think is they actually don't interact very much at all in terms of the microbial communities you find in one versus the other. And I'll show you a little bit of evidence of that in the next slide. So once again, this is very overview. It's probably more information than I really should put on a slide, but basically it shows that if you look at the upstream river locations, which are on the far left, and then you compare the raw wastewater sources, they're very different, not surprisingly, but the community wastewater in terms of microbial diversity and the hospital waste from the two hospitals aren't really that much different each other. However, once you get to the influent of the treatment plant, which is here, it's starting to see the effects of having the wastewater go through the sewage ecosystem. So there's some selection going on relative to the changing and evolution of the new community that then arrives to the treatment plant. Now, the place where the biggest changes occur actually are in the treatment plant, but they change in a way that's different than what we used to believe. What comes in the influent is really a mirror of the wastewater that's coming to the treatment plant, and it's characteristic of what you see in the sewage. However, if you separate out the communities that are present in the floc phase versus the non-floc phase, say in an activated sludge system, the communities you find in those two places are completely different. And the floc phase is really dominated by organisms that actually like to flocculate. So it doesn't seem to be a particularly selective place for organisms to do with resistance. And in fact, resistance diversity, as I indicated before, in floc is actually quite low. Resistance abundance is quite high because there's a lot of floc. But relative to mobile resistance, you find the resistance in the floc is actually quite low. And we've seen this in more than one location. So if you look at this, this is a network co-occurrence analysis of combining microbiomes and resistomes. And we look at the liquid phase that's going out, the liquid part of the clarifier, and the floc that's settling into the recirculation system in the treatment plant. We find that the huge majority of the mobile elements that are carrying resistance genes are associated with bacteria that are in the liquid phase. And that, in fact, in this treatment plant, because this is from the treatment plant in Spain, we found very, very few mobile elements that seem to be associated with resistance genes. We also didn't find many enteric strains, or ones that would presume to be enteric, prevalent in the floc either. So you've got two very, very different entities, the liquid versus the floc phase in the clarifier, and that has real significant implications relative to how you should operate a treatment plant to reduce resistance coming from the treatment plant. So when we first had this data, it was primarily in Spain, but in Darwin, which was our JPI AMR project with Bart Smets and Soren Sorensen and a number of others across Europe, we did equivalent studies to what we did in Spain, but we did them in Denmark, the UK, and in Spain. We redid the work in Spain. And what we found was something really interesting. And this is another one of these things that I think it sort of confirms our theory about the clarifier or the solid separation unit being important. If you looked at the wastewater, this is comparing microbiomes. You find that the sewage looks like sewage. Obviously, there's going to be differences in Denmark, the UK, and Spain on a local level, but generally speaking, they cluster in about the same way. And this is based on winter and summer samples. But if you look at the activated sludge, the floc phase, it varies dramatically based on the place. So Spain, Denmark, and the UK separate quite a lot. Now, we initially thought that was because they were different countries. But then we looked at it more carefully, and it was because they had different ways of solid separation. So what we've come to believe is that what you get in terms of the liquid phase effluent and what you get within the floc relative to resistance genes and mobile genetic elements is very much controlled by the solid separation process in the system you're studying. Now, we've studied more systems than just the three within this study since then, but the data is corroboratory. So we don't see this universally true, but we see it largely true. So the question is, what controls mobile elements in the effluent? So I said in the liquid phase we seem to have more ARGs associated with mobile elements in the liquid phase than the flock phase. So we also see another observation that I didn't say is that we see a lot more enteric strains or pseudo presumed to be enteric strains in the liquid phase. And what that what struck us was that, okay, if you think about E. coli and a lot of gut organisms, they're not organisms that are intrinsically flocculators. And I think there's 100 years of microbiology that confirm that, because we use E. coli as our lab rat for microbiology because we can measure it by optical density. And so that means it doesn't form particles that become difficult to use optical methods for quantifying the abundance of what you've got. So E. coli and organisms similar to E. coli tend not to associate naturally with flock. And if they're the organisms that are predominantly carrying the resistance genes from the original source, i.e. the gut that's going down the sewer eventually to the treatment plant, they're going to go through the liquid phase and not associated with the flock. We also see the flock and the liquid phase being very, very different in terms of their microbiomes, which I showed you earlier. And what this has led us to believe is you can't look at the effluents from a treatment plant by combining a sample, by using a sample, say, from an activated sludge unit. Because what it does is it's combining two very different ecosystems. You're dealing with flock ecosystem and the liquid phase ecosystem. If you look at the resistome and microbiome of them combined together, you get a completely misrepresentative picture of where the resistance genes are and where the mobile elements are. Because one in the liquid phase is where you predominantly find most of the mobile genes, and the solid phase is where you don't. And that you don't tell that if you measure the two things at the same time. So what we've come to conclude is that the flock itself is an ecosystem that's unique, and it's not controlled by anything related to AMR. It's controlled by the tendency of organisms to flocculate. And in fact, if you look at the microbiomes of flock, it's dominated by organisms that are more surface associated, organisms that like to flocculate. And as a consequence, it's not surprising that we don't find a high abundance of mobile resistance associated with the flock phase. However, what that then says is the part of the liquid, of the clarifier ecosystem we care about is what's going out in the liquid, because it's carrying the organisms that don't flocculate, and they're the ones that are most likely to be associated with the gut someplace upstream. Now, there's some other things about the sludge and the flock that are really important that I think for understanding this, and this is important relative to monitoring, and this is what I call the fallacy of clinical class 1 integrons. First, I just want to make a general comment about hospital waste. This talk's not about hospital waste, but one of the characteristic differences we find between hospital waste and community waste is the relative propensity of clinical integrons. And there's higher abundances of clinical class 1 integrons, but also those clinical class 1 integrons are carrying somewhere between 5 and 10 times more resistance genes per cell than if you see that from the community. Now, as those organisms integrate with the community waste, sometimes you don't even see that effect of the treatment plant. But there definitely is a difference in the abundance and mobility of resistance genes that are coming out of hospital waste. However, the thing that we've discovered most recently that I think is really cool is because of this worry about the difference between sludge and the liquid phase, we were worried about what integrons were actually telling us. And so what we did was we developed a more refined suite of gene probes, which we haven't published yet, that this works under review at Environment International, which allow us to segregate out clinical integrons, non-clinical integrons, and empty integron cassettes. And so what that tells us is, okay, which integron is actually carrying a gene that might be resistant and which ones are carrying nothing and are empty shells. And the two places where we find the empty shells are in the activated flock and also in the downstream river receiving water environment. So what that does is it reconfirms the fact that you're getting flock doesn't necessarily mean it's a selective for resistance. You might have a lot of organisms, but they're not carrying mobile resistance. And in fact, many of the cassettes, and we found one treatment plant where 70% of the resistance of the integrons were empty, uh, are not carrying resistance at all. Now, we still don't completely understand what that means, but we think it's something that certainly puts into question using something like a clinical class 1 integron as a marker for the spread of resistance genes, when in some places, the majority of those integrons aren't carrying any resistance gene at all. So, to finish, I'm going to talk just a little bit about horizontal gene transfer, because I've made an observation through working in various different countries around the world that the greatest evidence of horizontal gene transfer to do with environmental resistance was in highly polluted oxygen-free rivers. And so my idea was that if we could develop a tagged plasmid assay with a genetically modified E. coli in this case, we could potentially look at the relative influence of redox and other factors on gene transfer in different types of environments. And so we set up a series of model wastewater ecosystems and did plasmid labeled tagged experiments, fate experiments. Now we also, just out of curiosity, we also looked at food web effects, which ended up telling us something rather interesting as well. So I'm not going to talk about the details of the assay, but basically what we did was we isolated an E. coli from one of our wastewater treatment plants in Newcastle. We genetically altered it and made it constitutive nalidixic resistant. And then we provided it a INC-P plasmid carrying 3 different types of resistance genes. And then we fluorescently labeled the host. We fluorescently labeled the plasmid, and then we had the nalidixic acid resistant to try and recover the host. So we took those genetically modified organisms, and we added them to bioreactors that had been maintained for over 6 months at low redox, at aerobic conditions, mid-redox conditions, which were denitrifying, and anaerobic conditions, which were highly reduced. And then we looked at the fate of our plasmid and our host in those systems. Just to give credit where credit's due, this was based on the assay that Soren Sorensen developed, and then Bart Smets modified using the filter assays. However, we developed the assay in liquid phase instead. So what we saw superficially was our host disappeared very rapidly in the aerobic reactors, but it didn't disappear rapidly in our anoxic or anaerobic reactors. And this was equally seen in the bulk liquid as well as the sponge biofilm. So what we did was we harvested transconjugants. We just chose roughly 60 from our aerobic, anoxic, and anaerobic environments. These are ones that we've grown on plates. And we pulled out 39 stable, what we call stable transconjugants from that pool. Now, this is where things get kind of interesting, and it sort of maybe fits into work that Rebecca might be talking about later, is that we found the predominant locations where the transconjugants were occurring were in biofilm. And particularly, they were in anaerobic biofilm. What was interesting, however, was that we only found E. coli that were transconjugate. Now, that might be because we biased the system because of the plasmid we used and the fact that we started with an E. coli. But we found all of our transconjugants all of our transconjugants were E. coli, and the lion's share of them were actually from anaerobic biofilm. It doesn't say it's statistically significant, but it says that there's a strong tendency towards. However, that didn't explain the reason why we saw differences in the abundance of the host. And just as a matter of curiosity, I had our technician do protozoan counts in our different bioreactors in the period surrounding the addition of when we provided our fluorescently labeled donor. And what we found was that soon after we added our donor, we saw a slight increase in the abundance of our protozoa, which we assumed was an ecological consequence of the fact that we provided it more food. They basically were protozoa prey. which slowly declined over time. This happened both in the biofilms and also in the liquid phase in the reactors. And what was curious is that we found that the protozoa in those reactors were glowing, presuming that they were eating our fluorescently labeled tagged organisms, and they were becoming fluorescent as a consequence of their food. We can't prove that absolutely, but we think it's strongly suggested. So, final thoughts. First, I strongly encourage people to think about wastewater networks as a whole series of discrete ecosystems that contain sub-ecosystems. And everywhere in the network is different. Some parts of the network, you're going to see high levels of gene transfer. Some places, you're going to see no association at all with antibiotic resistance or mobile elements. And what we're trying to do is determine which parts of the network are the parts we should be focusing on to understand resistance gene transfer. But what we know from the little bit that I've told you today is that what drives resistance gene and mobile element removal is not straightforward. If you look at liquid wastewater treatment plant effluents, and we've seen this in multiple treatment plants now, but I've only shown you 3 here. The liquid phase have strains that are mostly related to the wastewater sources, including enteric strains that come from the original source, which might be the gut of a patient in the hospital or the individual in the community. And those strains tend to carry more mobile resistance genes, and as a consequence, if we're thinking about removing the resistance genes that might spread in the environment, what we should really be doing is trying to remove the non-soluble biosolids fraction through our separation system, the clarifiers. Obviously, now that's going to suggest maybe using membrane reactors, but we should be focusing very heavily, if we want to protect the environment, on reducing the non-soluble biosolids fraction. The fallacy, I think, is that the flock phase may be less significant relative to resistance gene transfer and horizontal gene transfer. And we see relatively low genetic mobility. And in fact, there are places where you find many of the integrons empty. So our final advice is that biosolids removal is key for reducing the spread. And it's especially true if you use anaerobic processes, because it seems like horizontal gene transfer is more inclined to happen under those conditions. Thank you.
Many thanks, David, for this really interesting talk. Thanks for giving us an insight in the diverse ecologies of the diverse different networks. Is there time for a question on this one?
Yeah, we have a few questions coming in. First one is from George Southberg, and he's asking if the composition in the liquid of the activated sludge is the same as in the effluent. So did you specifically look at the sludge without the water? So did you dry or remove the water from the sludge?
Yes. Yes. So what I was showing you was the flock itself, and then the liquid phase that was surrounding the flock. And so that was when we discovered that the flock was so much different than what was in the liquid phase. What we actually found was the influent to the treatment plant bared quite a lot of resemblance to the effluent that was going out of in the liquid phase of the clarifier. So it seems to be a pool of bacteria that are in the wastewater that are going through the treatment plant functionally without treatment. So they're just going through because they're not associating with the biosolids in the biological treatment unit. Now, that suggests certain types of treatment technologies are going to be better than others relative to removing resistant bacteria because of it, especially if they're non-flocculating strains. But the answer is yes. We separate out the liquid from the flock phase and did the comparisons between the two, and they're very different.
Yeah. One last one.
One last one. Okay, so I'll then take this one. The question is, what kind of technology of wastewater could be efficient enough for the removal of these resistant genes?
Oh, That's the great question. And unfortunately, the bad news is for water companies. I don't really think. I think the best way is by using membrane-based separation systems if you really want to do it. That basically, I was going to show in the talk, but I thought it would be too long. But we've studied all different types of technologies, and membrane bioreactors remove resistance genes 2 to 3 log better than any other treatment technology. I'm not that keen on using advanced oxidation methods, except when the waste is very small volume because it becomes too expensive. The problem is the membrane bioreactors, which are really effective, are also very expensive. And so if you really want to remove resistance genes, you really have to take, as I said, you need to remove the non-settleable fraction of what's in the effluent. But that's not going to be cheap, and therefore I think we've got to be very conscious of the economy of that, and so although it's the best way, maybe it's not the most economical.
One more question then. This is from Professor London, and it's what about the combined sewer overflows? Do you think it is a relevant source of antimicrobial resistance?
Absolutely. And I mean, we in one of the studies we did, this was in another place in Spain where there was combined sewers, that the effluent that was coming from the treatment plant had much lower levels of resistance than were in the sediment and the water in the river to which the treatment plant was feeding waste. And we first, that didn't make any sense because we didn't think, we've never seen much resistance growth in the environment. But what we discovered was they had a wastewater bypass. And with when we had the data, it was during the winter when there was much more rain. And what we were measuring in the downstream side of the waste treatment plant was the cumulative effect of stormwater overflow, basically bypassed water around the treatment plant. And so it was getting wastewater directly. And so it's an excellent question. It argues very strongly for not using combined sewers, but many parts of the world, including the UK, that's just the way it is. I'm from Canada, and you don't see combined sewers there, and it's a different scenario. But that's an excellent question. It's probably the hardest one to deal with from an operational perspective from the water company.
Very much so. And I think these are two excellent leads for our practitioners from the water boards. So we have two questions for them to answer. What about the non-settleable biosolids, how to remove them in practice, and also the sewage overflows? But before we address that, we also have a lead for the next presentation, which is by Rebecca Palaris, because she also tried to tackle the question on which techniques within the conventional wastewater treatment plant might actually contribute most to the removal of resistance in the aqueous phase to the effluent. So it's my pleasure after 4 or 5 years working with Rebecca now to introduce her as the second speaker in this webinar, and to show us her results from the PhD study that she conducted at Wetsus in Leeuwarden under supervision of Mark van Loosdrecht at the Technical University of Delft. Rebecca, floor is yours.
Thank you very much. Can you hear me well?
Yes.
Okay. So I will share my screen. Okay. So welcome all. My name is Rebecca Pallares Vega, and as Heike says, I'm a PhD candidate of TU Delft and Wetsus. And today I want to talk about the determinants of presence and removal of antibiotic resistance in wastewater. Well, Professor Graham already explained a bit about antibiotic resistance. I just want to mention that resistance is a growing global problem because, first of all, for all the antibiotics that are developed, some bacteria have become already resistant to them. So that means that in the near future, some of the infectious diseases that are currently being treatable won't be treatable anymore. Professor Graham also has point out that this is not only a problem on the clinics, that this is a multidimensional problem. We know that human and animals, they ingest antibiotics. Some bacteria within their gut might become resistant, and together with the feces, they will end up in the environment. And also, eventually, human or animals can become in contact with resistant bacteria that are in the environment. So there is a multidimensional interconnection. We study antibiotic resistance in wastewater treatment plants because these are the facilities that are connecting our anthropogenic or human-related antibiotic resistance and the environment. Wastewater treatment plants, they collect the sewage that contains, for instance, fecal bacteria, including resistant bacteria, and also other substances such as, for instance, antibiotics or biocides. And because of the characteristics of the wastewater treatment plant, it's suggested that they could be hotspots for the development and spread of antibiotic resistance, because we have these antibiotics that might be selecting for certain bacteria that are resistant, and also because of the high bacterial density within the treatment plant, there is also suggested that they might facilitate the horizontal gene transfer of genes. So today I'm going to explain you a bit the results that we obtained in our study, and then we can evaluate if this statement is indeed still standing in our case. I'm going to talk today about the results that we obtained in two studies that we did across the Netherlands. One was a cross-sectional study in which we sampled many wastewater treatment plants across the whole country, but only one time. And the other one is a long-term study that we did in 3 of the treatment plants with different kind of treatments, and we sampled it monthly throughout a year. In both cases, we investigated resistant genes, or what we call ARGs, by qPCR. And also we investigated 2 mobile genetic elements that are associated with antibiotic resistance. And to track the amount of bacteria, what we call bacterial community, we also measure 16S RNA. The first question we want to answer was to know what was the occurrence of removal of antibiotic resistant genes. But also, we wanted to go a bit further and study which factors were affecting the incoming levels of resistant genes and also the removal capacity of the plants. So now I'm going to introduce you a bit the results. First of all, what is incoming to the wastewater treatment plant? Which genes do we have from the ones we selected? We could detect all. Here we have the total community, 16S. These are the resistant genes. I don't know if you can see my pointer. Here we have the resistant genes, and here we have the mobile genetic element. And just to tell you briefly that in the Influent, we have resistant genes in levels from 10 to the 4th to 10 to the 7th gene copies per milliliter of Influent. Some genes were indeed more prevalent, like for instance RMB, that is for erythromycin resistance, or sulfonamide resistance, SUL1, and others that are, for instance, for more advanced antibiotics, or antibiotics that are used maybe when you're a bit more sick, like beta-lactams, that they were less prevalent. But let's go now to the effluent and see how this looked like after the treatment. And then what we see is that for all the genes that we selected, there was indeed a noticeable reduction. And how much was this? So for the resistant genes, the removal was around 1.8 to 2.6 logs, and this translated into percentage of removal is from 98 to 99.7%. So also when we do statistics to see if this removal is significant, we can say that yes, that wastewater treatment plants significantly reduce the absolute abundance of resistant genes. Yet, it's important to remember that even though we have this important removal, wastewater treatment plants in the Netherlands, they are still discharging from 10 to the 4 to 10 to the 6 gene copies of resistant genes per liter. And that accounting for a plant that is medium or large, around 100,000 cubic meters per day, means that they discharge from 10 to the 12 to 10 to the 14 gene copies of resistant genes per day. Well, we know that wastewater treatment plants, they reduce this total amount of genes, but they are also proposed as selectors, places where they can select the resistance, and also that they eliminate a bit better the bacteria that are not resistant, that bacteria that are resistant, and then in the end, the relative abundance of resistant bacteria may be increased. But actually, we don't see this in our results. In our results, what we see is that there was either a small or a reduction or no change in the relative abundance of the genes. And the relative abundance is the genes divided by the total amount of bacteria. So we don't see an increase, but we see a decrease. And the exception to this was to this gene here that I haven't referred to it before. That is the Corby gene that stands for MP plasmids. That, for instance, are the kind of plasmids that Professor Graham has used for horizontal gene transfer, are the ones that are normally used for this kind of experiments to see the exchange of resistant genes in between bacteria. Okay, so now we already know how much we got into the plant and how much we could reduce, but we said that we wanted to study also which factors may affect the incoming levels of bacteria towards the plant and also the removal of these bacteria. No? So we first will go to the influent factors and study if, for instance, the presence of healthcare or the temperature or the flow were affecting the incoming levels. And first of all, I want to say that, well, we don't observe that when there is a hospital or nursing home in the catchment area, there are more higher concentration of resistant genes coming into the plant. It's important to mention that indeed hospitals and nursing homes are places where there is the sewage that produce these places. They have higher occurrence of antibiotic resistant genes than normal community, and also this quantity of resistant genes is more diverse. There are more number of different genes, but because they represent just like a 1 to 2% of the total volume of water that comes to the sewage, actually they don't contribute so much in the total numbers. So we don't see an effect when it comes to the influent. Regarding the temperature, well, we know that sometimes it's said that in some seasons there might be more prescription of antibiotics, so there may be more resistant genes going around. Well, in this case, we didn't study season, we studied temperature-wise, and we observed that indeed in colder temperatures, there were a bit higher amount of resistant genes, although significant, but indeed the effect was moderate. And what is maybe curious is that the resistant genes were higher when it was cold weather, but the E. coli was the other way around, was when it was warm weather. So it was a bit different. Maybe a bit more interested is one we found for flow. So we wanted to know if, for instance, the concentration of resistant genes were changing with the flow. If we have a stable community of resistant bacteria within a city, citizens that they discharge always the same amount of resistant bacteria, if we have a normal flow, maybe we will have, for instance, 4 gene copies per milliliter. And if there is rainfall, then this should increase. They should be diluted because the flow increases, no? To measure this effect across all the plants that they have different flows, we have developed a new term that we call hydraulic load factor. That is the amount of flow that the plant is processing the day we sample, divided by the average amount that the plant is treating. And this is a way to normalize the flow across all the plants that we have sampled. So what did we observe when we saw flow and concentration? Well, we didn't see a really clear pattern across the genes. So there was not a clear tendency of dilution or increasing. It was dependent of the gene. Instead of concentration, we said, okay, we are going to measure loads, because loads are the concentration times the flow. And therefore, it doesn't matter that one day I will have a more concentrated stream or a more diluted stream, that if my bacteria only come from this fecal source, in the end of the day, I will always have the same, because I will multiply my concentration times the flow. Therefore, if we hypothesize that this is the case, if we increase the hydraulic load factor, we will always have a constant amount of load. But actually, what we observe is not that. We observe that there is an increase in the loads when there is an increase in the hydraulic load factor. And that means that there are other sources of antibiotic resistant genes besides fecal matter. And we propose that these other sources are biofilm that accumulate within the pipes and that is washed out within the period of high flow. So we already have talked about the pattern, the factors that may affect the incoming concentration of resistant genes, and now we will go to the ones that may affect the removal. And I must say that despite we study many of them, most of them didn't matter for the removal of genes. So for instance, it didn't matter that our plant had a primary clarification step or not, because apparently this step is not really helping to remove resistant genes. It also didn't matter if we had a system with a chemical phosphorus removal or biological phosphorus removal. There was also no difference, or the type of sludge, for instance, floccular or granular, we didn't see a significant difference. But what we saw a difference in was, again, flow. With a higher flow, the removal of genes was worst. This was an homogeneous effect for all the genes, although the intensity varied per gene. On average, every time the flow was double, our removal capacity on the plants was reduced by 0.4 logs, although for some genes, like for instance quinolone S or RMB, this effect was even worse, and we lose removal capacity by 0.8 logs every time the flow was double. We also observed the same effect for E. coli, even a strong effect than for the genes. And then we hypothesize that this is caused because the water spends less time in the biological treatment than it should. And you might say, yeah, but before you said there was no effect, for instance, on the hydraulic retention time, you didn't see that on the factors that you tested. And it's true, there is no, apparently there is not a single hydraulic retention time that is best to remove genes. But what we observe is that probably each plant has its own retention time, that is hydraulic retention time, that is optimized for this plant to remove the normal pollutants, the COD, the nitrogen, the phosphorus, and probably also the genes, where it works the best. And when there is higher flow, this is disturbed, so the plant doesn't perform well because the water doesn't spend the time that it needs in the biological treatment. Still, with the flow, we cannot explain all the variability that we see across plants. So we thought to study other parameters, like for instance, the temperature that we also observe in the influent, that it was having an effect to know if this variability was caused by these other parameters. And we know that temperature that varies across seasons affects the microbial community within the activated sludge, and therefore it might also affect the removal capacity. But actually, we don't see this effect. It doesn't matter the temperature in the ambient. We didn't observe a strong effect or a significant effect in the removal capacity of the wastewater treatment plant. Contrarily, what we observe, and this is a bit in connection with what Professor Graham said, is what there is a higher amount of total solids in the effluent. We observe that is correlated also with a decrease in the removal capacity. And of course, the effect is moderate as per unit of turbidity, but we observe that there is an effect. So now I want to just mention a bit quickly other part of the work we have done. We have said that part of this mantra that is repeated, that horizontal, that wastewater treatment plants are hotspots for antibiotic resistance, is also connected to the fact that they are thought to be places where horizontal gene transfer can happen often. The problem is that actually, although we know a lot about conjugation, that is the exchange of plasmids, in this case that contain resistant genes, and it's a kind of horizontal gene transfer. We know a lot of that because there's a lot of studies. Most of these studies are done in vitro and also under optimal conditions that are not really relevant, for instance, for the case of the Netherlands, for instance, in temperature or some. So we wanted to study some small parts, like some small experiments we did about, okay, if we take a small assay, easy assay, and we put some conditions that go from the optimal, that we know it works, towards the more environmental, what we observe, is it affected, this transfer? And actually for temperature, we see a small decrease, for instance, when there is lower temperature in the amount of transfer, but the biggest influence we observe it for nutrients. So we think that different nutrients that in the environmental conditions is tend to be lower than in the optimal conditions, then this is a parameter that is indeed affecting the conjugation. Of course, doing these experiments in the lab is not ideal, and the ideal thing would be to do it in the field. And a middle term is to do experiments in microcosms that we call in situ. And actually, we could do some approach, preliminary approach with together with the University of Copenhagen. And just to quickly tell you that we investigated if indeed we could measure directly transfer, and then without any other culturing step, just to measure if we could recover these transconjugants. And actually, indeed, we could do it. If we see here, we could recover some transconjugants, but they normally occur in low amount. And when we move from, for instance, a nice temperature for our donor that was E. coli at 30 degrees, when we move to 15 degrees, or when we move to um, other conditions, then we see that our transconjugants, when we detect them, they are really within the limit of quantification. So therefore, I want to say that yes, we could detect it. We can detect the plasmid transfer in situ, but still we need to work around the method. And with that, I would like to arrive to the take-home. And my take-home message in connection with this first part that we saw on the wastewater treatment plants hotspots for horizontal genes for antimicrobial resistance, I would say in the Netherlands, at least, wastewater treatment plants help to mitigate the spread of antibiotic resistance because they remove antibiotic resistant genes. And just some summary of the things we observed. For the presence, we observe a bit higher amount of genes when there was cold temperature. And also what we observe is that flow is an important parameter, but it's true that in the influent, it's better to use loads to assess the effect of flow. And also to measure, for instance, temperature, we recommend everybody to use loads because you already include the information about flow. And by using loads, we know that there are we hypothesize that there are other sources of antibiotic resistant genes coming into the wastewater treatment plants, and we propose that these are from sewer biofilms. For the removal, very important, our plants, as I said, they remove genes, they remove up to 99.7% of the genes, but still they release a portion, 10 to the 4 to 10 to the 6. So this is still something to improve. There is no obvious better design and operational condition for the plant, but we know that when there is an increase in flow, the plant does worse in removing resistant genes. And last of all, that effluent turbidity is also worse correlated with a worse removal of resistant genes. And finally, just to say that, well, we could measure horizontal gene transfer in situ, and it's promising, but it needs a further method optimization. And with that, I would like to thank everybody that has been part of this work, to the wastewater treatment plant operators that helped me a lot during the sampling collection, to my students also that did part of this work, and for the part of the horizontal gene transfer, special thanks to, of course, the colleagues in Copenhagen University and to Gonzalo Macedo that did part of this work too. And with that, I would like to thank you for your attention, and I'm ready for your questions.
Many thanks, Rebecca, for your very clear talk. I'm sure there are many questions. I'm also sure that we will have to work on the weather in the Netherlands, so we have to make it stop raining. So that is very clear. I'm not so sure about temperature because temperature seems to have mixed effects on conjugation and on presence of genes, so that's not that clear. Lucia, is there an urgent question to Rebecca?
I would like to pose this question from Joana Abreu Silva. Rebecca, which was the criteria for the selection of ARGs and genetic elements? And congratulations for your work.
Yeah, so we decided to choose these genes because first, they are related to antibiotics that are currently used in the Netherlands. And also there is always problems to harmonize results because a lot of people that's different. So there were some guidelines like 2015 from consortium of people that work in AMR, and they suggested a list of genes, and these are also included in this list. They are not all from the list, but they are a great part of the list. And as I said, also because they are relevant for the antibiotics that are in use in the Netherlands. And then for the mobile genetic elements, we chose integron because it's commonly associated with with several resistant genes, and also MP plasmids, because as we said, are the common plasmid that are used for horizontal gene transfer.
Thank you. For the sake of leaving some room for the general discussion, we would like to move on to our last speaker, Maarten Nederlof, who is a senior water technologist with the Dutch water board AMR. And from there, he's also involved in the Dutch Ketenaanpak medicijnresten, or chain approach to reduce pharmaceuticals in water, where he's busy with implementation of full-scale advanced treatment plants. And I hope, Maarten, that you enlighten us on the challenge to remove the non-settleable fraction and to reduce sewage overflows. Large challenges ahead.
Yeah. Thank you very much, Heike, and indeed for inviting me to this beautiful studio in Leeuwarden. So, just a few words maybe on the rainwater part, because that's not part of my presentation. So the challenge is to separate the rainwater from the wastewater in the Netherlands, and the municipalities are busy doing that, but it will take some decades before we reach some serious percentages of separation. So it's a going on, but still takes some time because all the sewer pipes are already there. So it will take some time. But starting with my presentation, I'm indeed Maarten Nederlof, working at a water board. So it's not a scientific presentation, but more about things that are going on in the field of wastewater treatment plants in the Netherlands. And I will talk about the next step, and that is investing in what they call quaternary treatment steps, or in Dutch, de vierde trap. I can do this myself. So a brief outline of my presentation, few words about the European and Dutch legislation. And the conclusion is there is no standard for pharmaceuticals yet. But they are there. We measure them in urban wastewater and even in surface water and even in drinking water. So there is a need for a Dutch approach, in beautiful Dutch, de Ketenaanpak. We use technologies. So the first installations that are being implemented use, of course, the proven technologies that have been studied years before. But there are also some new promising technologies, and talking about the biosolids and the genes, so new words that I'm learning today. Maybe we have to look more at the promising technologies to what they do in addition to the present technologies. So maybe I come to that when I talk about the bigger picture. And the final question, and it's still not addressed yet, is there a need for disinfection or AMR reduction? So the word risk I didn't hear yet. So what is the risk for the environment? What is the risk for the public health? So maybe that's part of the broad discussion afterwards. So there are two European directives that are relevant for wastewater treatment. And it is the Water Framework Directive that's dealing with the surface water quality. So in 2027, there should be a chemical and an ecological good water quality. The surface water should be suitable as a drinking water source, so that you can treat surface water with simple techniques, whatever they may be, to make drinking water out of it. And the quality parameters are mainly focusing on nutrients on the one hand. So a lot of waterworks are busy removing them. And what are called priority compounds, and that are really toxic substances, and there are no pharmaceuticals between them. But there's some debate going on if some pharmaceuticals, such as diclofenac, a famous painkiller, I think, should be in that series of priority compounds. The other one is the Urban Water Directive, and that's really dealing with the wastewater treatment plants themselves. So it asks to use best available technologies to treat the water. There are standards for nutrients, and there is an evaluation going on right now if the directive should be changed to include pharmaceuticals as well. In contrast to drinking water legislation, there is no additional legislation for wastewater treatment in the Netherlands. So in the Netherlands, we just follow the European directives. So what we are doing in practice these years are just implementing measures to fulfill the requirements of the Water Framework Directive that should be done before 2027. That is, on the one hand, the water management, so spatial planning, planting trees, creating the right water velocity, and on the other hand, improving the water quality. And there are different sources of contaminants, of course. So agriculture delivers a lot of nutrients, but the wastewater treatment plant, of course, as well. And the challenge is to illustrate that we are quite busy with that from the present situation, we have to reduce the concentrations of nutrients by a factor of 4 to 5. So that will result in some extra treatment steps. And the question is, of course, when we start removing the nutrients, is that an opportunity to combine it with the removal of pharmaceuticals or even AMR? I'm just learning today, as I told you. On the organic micropollutants, that's the topic I'm dealing with. There's thousands of different substances there from different sources. So we focus on this moment on pharmaceuticals because we more or less know who they are and what they do. But there are, of course, even in domestic wastewater, other organic micropollutants such as personal care products. I think a few years ago, even Wetsus studied them, as I remember. So the pesticides is a famous group of organic contaminants, and there are more and more attention for industrial wastewater with really nasty compounds that really bothers the drinking water production. So one of few challenges to present is that not all those components can be analyzed in a lab. There's not for all components, an indication of the toxicity. So there are no drinking water standards for individual pharmaceuticals. And there are some PNECs available that are predicted no effect concentrations for the environment. So we are busy with what are the critical parameters and the corresponding sources to evaluate what measures we have to take. And one of the discussions is, of course, could we do something at the source, so prevent contaminants from coming into the wastewater. For some compounds, that is a good idea. Diclofenac, for instance, could be reduced, but a lot of pharmaceuticals keep us healthy. Because we are not saying that we are forbidden to use any pharmaceuticals at all. So they will be there, and we have to do something with it. So as I told, there's no regulation yet. So we are just doing some things, and water boards, at least some, take their own responsibility because they are there. They might have some effects. So we are busy with additional treatment steps. Some examples of concentrations. I just picked 3 pharmaceuticals. A famous painkiller, diclofenac, metformin, which is a diabetes pharmaceutical, and clarithromycin, which is an antibiotic. I thought it should fit in the program of today. And just to illustrate that the concentrations in wastewater really differ. So metformin is found in very large concentrations. It's no typing error, so it doesn't play with the comma in my tables. And diclofenac and clarithromycin are found in lower concentrations. The good news is that the wastewater treatment plant removes part of the pharmaceuticals, especially metformin is well biologically degradable, so a very high removal percentage. When we compare surface water concentrations upstreams and downstreams of the wastewater treatment plant, you see an increase. So we are really adding something to the surface water, so the concentration increase. Even in the Maas, which is a drinking water source, low concentrations of pharmaceuticals are found. And to be honest, even in drinking water, and I'm allowed to present this today, even small amounts of, for instance, metformin are found in the drinking water. So that's the situation. So the PNECs differ quite. So the PNECs for diclofenac and clarithromycin, always different words, are very low. And metformin, luckily, has very high concentration, but also the PNEC is very high. So depending on the different pharmaceutical, the risks are very different. There is a debate going on, is there a relation with human toxicity? I don't think so, because medicines are made to make people better, so it would be strange when they are toxic for humans, but still they don't belong in the drinking water, and therefore there are some target values. So the European River Memorandum has a target value of 0.1 microgram per liter, which is really low, and an indicator parameter for the drinking water directive in the Netherlands is 1 microgram. So that is the range of concentrations that are found. So this is the Dutch approach, the chain approach, pharmaceuticals in water with 3 parts. So we are not focusing on wastewater treatment alone, but we start at the very beginning of the development of new pharmaceuticals that might be less toxic for the aquatic environment. A lot of work has to be done in the description and use. Just prescribe the amounts that are really necessary for the patient and not more. And of course, there will be some waste. And when you have packages of pharmaceuticals in your house, please bring them back to the pharmacy and prevent pharmaceuticals in the wastewater as much as possible. A few years ago, the STOA already mentioned did a hotspot analysis, and it's a different hotspot than in the previous presentations. So these are the wastewater treatment plants that might have a contribution to the concentration of pharmaceuticals downstream the wastewater treatment plants. We have more than 300 wastewater treatment plants in the Netherlands, and about 100 are considered as a hotspot, that they really have a significant increase in concentration. So all the yellow dots in the picture are less relevant, and the blue dots that are more or less the hotspots. And then the water boards consider, it is a model calculation, so what is really going on in our region. So this is an example of the Maas River region. So we did additional water quality measurements to confirm whether the pharmaceuticals are indeed present in the surface water. And of course, when you discharge your effluent in a river, what are the functions of those rivers? So that has a function as an aquatic ecosystem. There's not an ecosystem David and Rebecca told, but a natural ecosystem to make the distinction. And the Maas is used as a drinking water source. So that's the reason for us to pick some of the hotspots to install an installation. I'm happy to be a program manager of the national program, which intends to speed up implementation of new technologies. So the first tranche is going on right now with 14 wastewater treatment plants installing an additional technique, 14 of the about 100. So it's a start. Still a few to go, I would say, with proven technologies like ozone oxidation and powdered activated carbon dosage. And it is partly financed by the Ministry of Infrastructure and Water Management. So at the water board AMRs, where I'm working, we have 7 wastewater treatment plants in total, and we are considering to build 3 additional steps at Ooyen, Dintag, and a third location we didn't decide yet before 2027. We are not focusing on one technology, so depending on the situation, depending on the state of technology, we choose a technology that we like. So that might be powdered activated carbon, but it might also be ozone or even a combination of it. So so this is the one word I didn't translate. Sorry for that. So the protocol in good Dutch is powdered activated carbon. So the removal of activated carbon is quite well. We think that ozone does the job a bit better. And of course, in the practice, in the real world, also costs, energy consumption, and the formation of byproducts are relevant aspects, especially when with oxidation with ozone, there is bromate formed, and bromate is a toxic component with a rather severe drinking water standard. So solving the one problem could cause the other. So think about what you're doing. So one of the challenges is to keep the bromate formation as low as possible. And as I said, the combination of two technologies might be attractive to remove a broad range of different contaminants, because all those organic micropollutants have very different characteristics. So one picture of a colleague of mine doing the pilots. So we don't believe things from abroad in the Netherlands. So we do our pilots ourselves. So we did at ALR Rixel a pilot study with ozone and UV in combination with hydrogen peroxide. And it turned out that the energy consumption, in contrast to what we expected with UV, was much higher than we thought before. So we were glad to have done some pilots to choose the right technology. Ozone had a problem that we indeed got a very significant bromate concentration. So the conclusion was a bit disappointing. So the perfect technology is still not there. So we are still looking for the right technology. Some ideas on the removal. Luckily, the wastewater treatment plant itself, which is a biological process, removes some of the pharmaceuticals quite well. So as I told, metformin is removed for more than 95%. So it's rather biological degradable. Other compounds is less removed, like diclofenac and clarithromycin. PACAS does the job very well. It's the powdered activated carbon. And for most components, ozone is a bit better. But there are compounds that are removed very well with ozone and less with powdered activated carbon, and vice versa. So maybe some promising technologies, and David mentioned already the membrane filtration. So maybe that's technology for the future to remove, as I remember, the biosolids and the genes. So the membrane filtration might do that job in combination with the removal of the pharmaceuticals. So that might be a nice idea. But depending on the type of membrane filtration, you get a concentrate problem, so that you get concentrated salt solutions, and that's a new challenge to deal with. So the main message of my talk is that solving the one problem often causes a new problem, and you have to ask, what are we doing, and is it a better result than we had? There's a lot of interest in artificial wetlands. So instead of installing all kind of fancy technologies, could plants and roots of plants do the job cheaper and as well as the advanced technologies? I put a question mark there. So the other ones are the nanoadsorbents as an alternative for powdered activated carbon. And quite promising is, amongst others, the zeolites, because they can absorb, when you have a mixture of zeolites, they can absorb the organic micropollutants quite well, and you can regenerate the zeolites with ozone. Some pictures of the nanofiltration. So as an example of the one direction is going to even more high tech, and the other direction is used natural, maybe the natural ecosystems to do the job. So make use of them. Maybe back to the bigger picture. So there's more to do than just removing organic micropollutants. So I think there is a need for effect assessment of additional treatment steps. So what is the problem and what is the result of the additional steps? So a lot of interest in bioassays. I thought that one of the previous webinars was on that topic. So that's a good thing, I think. There are a lot of components, so that it's impossible to analyze all the individual components. So there is a trend in measuring target and non-target screening, that in one spectrum you get a large number of compounds in one analysis. There are other challenges. We have still heavy metals in the surface water, especially zinc. Ammonia is a toxic compound, maybe more relevant than the pharmaceuticals. Microplastics is a topic, and of course, the topic of today, the pathogens and AMR. And to my surprise, I'm working in the field for 5 years now. There is almost nobody talking about removal of pathogens from wastewater treatment plants. I had a discussion with Heike a few years ago, I believe. But that's maybe nice for the discussion afterwards. So there is a trend, and that might be a very good opportunity to reuse the wastewater treatment effluent as part of a circular economy. So instead of throwing away the clean water, use it again, and then there are more opportunities to treat it in the correct way. But there are also restrictions. All the consumers have to pay the bill of the water board. That's still remains the case. We have to deal with the climate footprint. So we have to, as an ambition of a water board, we want to reduce our climate footprint. So there are limitations because any additional treatment steps increases the energy consumption. So maybe back to the regional validation, and I'm a tourist myself, so I'm happy to walk around the river Geul, which is a tributary of the Maas, or canoeing on the Dommel, and some people swim even in the Maas, and maybe that's a reason to reconsider our targets with respect to microbial quality. So question mark again there. And recently it was already mentioned, the COVID pandemic is a serious problem in whole of the world, but it helps us because it gives attention to the microbial quality of our wastewater and what's going through the wastewater treatment plant, and it helps us to put the topic on the agenda, I think, and that's a good thing. So, is there a need for disinfection? I'm happy to discuss that later on. So there might be, in cases where recreation takes place downstream a wastewater treatment plant, it might be a good thing. The reuse of wastewater treatment effluent, when people say we can make drinking water quality out of it, then also the microbial quality is one of the main issues, I think. And still, and I'm here at Wetsus, there is a need for more inside knowledge on the presence, increase, and risks of pathogens and AMR in the environment. Because I've worked at Wetsus in the past, I've learned that I should make a take-home message. So that is my last slide. So thinking of implementation of additional wastewater treatment steps, so the vierde trap of the quaternary treatment step. It's of course good to improve the water quality. Don't wait for the legislation. But as a colleague of mine said last week, when the legislation is there, the thinking stops. So keep an open mind for opportunities and limitations. And I think the answer is a fit for purpose treatment. Thank you very much.
What a nice take-home message. Thank you very much. And linked to that, I would actually like to pose some questions to David, because that also gives us the opportunity to compare the Netherlands with the UK. Because I'm not from the Netherlands, and I always admire the Dutch way of not stopping to think once there's legislation, but going their own ways. But we also heard that the water boards in the Netherlands, sometimes they take action without legislation, and they take their responsibility, and then they sometimes differ with respect to how much they do this or not.
Depends on the water board, of course. So the
Exactly.
water board has more ambitions than the other.
Exactly. How's that in the UK actually? What is the role of the local water facilities in safeguarding quality or in also pushing in developments to more advanced treatments?
I think it sounds like the system in the Netherlands and the UK are quite similar, and in the sense that in the UK there's 14 large water companies that handle water in different parts of the country, and some of the water companies are very, very proactive, and other ones are less proactive. So I think the water companies maybe are all a little bit prone to waiting until they're sort of obligated to do things, but some are pretty good about planning ahead. So even if rules change, they're ready to adapt to those changes. And so uh, but it varies from place to place. But generally, it doesn't sound like it's that much different.
But in England, there are private companies or not? Because in the Netherlands, it's still the government. So it's
in England, they're private companies.
Yeah.
But they're all regulated by a fairly stern board that is governmental. So they've got very strict practices, so principles for practice, both in protecting the client, which is the customers, but also in terms of protecting the environment. So they're private, but they're quite tightly regulated.
Maybe before we jump into a general discussion, what we could do is look at whether there are technical questions left for Maarten.
Yeah, there is one technical question. Have you been considering of using combined ozone and biofiltration technology for the removal of pharmaceuticals?
Yes, we did try that at Arle Rixel because one of the assumptions is that when using oxidation by ozone, you get byproducts that might be even more toxic than the original ones. So the discussion is going on whether you need a biofiltration afterwards to remove those byproducts. And there are some results that say that the effect is limited, and other results that say that the effects are there. So there's still point of research going on. So in my table, it was included.
Yeah. Shall I go for another question? This is again about, what do you think the public will say if we would have to install a next step against AMR after we have installed pharmaceutical removal?
After we installed pharmaceutical, so the seventh step or something like that, I can't count. I don't know. I think we have, as I see it, we have one chance now to do the right thing at once. So improving effluent quality is quite actual now. So in fact, because of the water framework directive, so the number of 2027, that's the reason that water boards are evaluating their effluent quality. And when you want to do something extra, I would say do it now. So in the same discussion, because I expect, or really I'm afraid that after 2030 or something like that, and that's very short in our view, because we make long-term plans, then it might be that climate footprint or circularity is more important than just improving the water quality. So the focus of the topics might change. So that would be my answer. So I find it interesting when choosing the right technology, and in those places that pathogens or AMR is relevant, then I would say do it at once.
That leads perfectly to my start of my discussion, because as you say, I think this is a unique chance now to implement the best technique for the questions we want to tackle, and that is a once in decades chance we have now. But if we now oppose the techniques that are now chosen, ozone treatment and activated carbon, with what we know about treatment efficiency of those techniques for both pathogens and AMR, David, you have already shown or mentioned that you also studied different techniques. What would be your answer on the choice of these techniques for removal of AMR in the Netherlands? Would these have been the techniques that you would have chosen?
Well, we've actually done, in the last couple of years, we've done pilot testing of different quaternary treatment options, post-secondary treatment in full-scale waste treatment plants in the UK in a number of locations. And we tried a number of the methods that Martin's mentioned. And so we looked at ozone, we looked at combined UV, hydrogen peroxide, we look at a number of options. And actually, our results are not that much different than his. And I'm not absolutely sure that these are worth the current investment. We found ozone with filtration as the best option for removing resistance genes. But we also found wetlands, especially in warmer environments, as a very effective way for reducing resistance genes as well. So I don't think there's an absolutely correct and incorrect answer. I think it depends upon the nature of the receiving environment, and so it's almost like a policy thing that as to what you would find acceptable and not find acceptable.
May I ask you a question, David?
Yes, go ahead. Yeah.
So the biosolids, is that removed by some kind of filtration? So we are installing rapid stent filtrations, for instance. Is that an opportunity to remove biosolids, or are they too small for that?